Okay, so what we need to do now is talk about, get a little bit more into the genes and proteins and how we go from this genetic code to actually producing proteins. And so we've talked about this a little bit, but we're going to go into a little bit more detail in this chapter. So remember, we've talked about the central dogma of biology, how there's information that is encoded on that DNA molecule based on the order that the bases are occurring in. That information is really valuable, so it stays in the nucleus where it's nice and protected. But in order for it to go out, it has to be transcribed into an RNA molecule, because those are cheaper copies that contain the same information. And then we can take that RNA molecule and translate it into a protein. And so that's the central dogma of biology. So basically, so you can write this in your notes: DNA, arrow with transcription over it, RNA, arrow, translation, protein. Okay, and just as a reminder, way back to chapter 3, when we were talking about biomolecules, our building blocks of our nucleic acids, so our DNA and our RNA, our nucleotides, we call these by their short names, which is A, C, G, T. And if we're talking about RNA, we're going to use U instead of T. And then our protein building blocks, those are our amino acids. So now that we remember all that, let's start talking about all this stuff. So what is transcription? Well, first of all, it takes place in the cell's nucleus. So we're copying the valuable DNA into a cheaper version that can get messed up, and that's fine, we don't care. So what happens is the DNA will kind of unzip, unwind that double helix a little bit. The base pairs will separate into this little thing that we call the transcription bubble, which is the cutest scientific term ever. And so in that transcription bubble, there's a polymerase that is able to create an mRNA molecule, one of our messenger RNAs. And let's just remember for a minute, what are our 3 major differences between DNA and RNA? So one, we remember that DNA is a double-stranded molecule, whereas RNA is usually just a single strand. The second thing that we need to remember is that there's a different sugar that's used in those nucleotide building blocks. In RNA, it's ribose, that's our 5-carbon sugar. In DNA, it's a modified version of that. It has an oxygen taken off, and so we call that our deoxyribose. That's why it's called DNA, because it's been deoxygenated. All right. So, and then the last and the third thing that's different between DNA and RNA that's going to be actually really important for what we're going to be talking about now is the bases. In DNA, we have A's, C's, G's, and T's. And then in RNA, we have A's, C's, G's, and U's. We don't use thymine, we use a different base called uracil. Okay, so that's going to become important when we're practicing transcription, which we're going to do here. So what we need to do is build a complementary mRNA version of this sequence, of this DNA sequence. And so how we would do this, we would say, okay, let's see, I think that G's and C's pair up together. So I would write a little C right here. Then I would say T's and A's go together. And I would continue this process, and then I would find this A, and I'm going to want to say, oh, A's and T's. I remember A's and T's, but this is where you got to be, this is where it gets tricky. RNA doesn't use thymine, it uses uracil. So instead of writing a T as the A's complement, you would write a U. And so this is what that looks like. So these things don't line up exactly, sorry about that. But we've got our G and C, our T and A, our G and C, our C and G, our A, and our U. And so just keep that in mind as you're going from DNA to RNA, that you're incorporating the correct bases. Okay, let's move along. So now that we've got our transcribed mRNA molecule. It leaves the nucleus by traveling through one of those nuclear pores, which is a little hole in the nuclear envelope. It goes into the cytoplasm and finds a ribosome. And now we remember that ribosomes are the organelles where proteins are synthesized. And so we look at this diagram, we've got our nice steps of the central dogma. We have our DNA, we've got the transcription bubble where that DNA molecule is opened up a little bit. We've got an RNA polymerase that is building our mRNA molecule. Once it is built, it leaves the nucleus, goes into the cytoplasm, and then finds the ribosome. And the ribosome reads, and with help of tRNA and rRNA, it creates this amino acid chain, which then becomes our proteins. Okay. So proteins, remember, we've talked about this a lot. Proteins are polymers of amino acids, and they're in biology. We have 20 common amino acids. There's a few others, but you know, they don't really show up that they're very often, and they're kind of the fringe cases. So we're just going to focus on these 20. And remember, the R group, which is indicated here in this little blue shading, this R means the rest of the molecule. And it's this side chain that gives the amino acids their chemical properties. And that's going to tell us how they're going to interact with the other amino acids, how they're going to interact with the aqueous environment, which is found in your cells. So some of these are going to be non-polar molecules. Some of them are going to be polar molecules. So they're going to have some that are hydrophilic and some that are hydrophobic. We're going to have some amino acids that are acidic, some that are basic, some that have a positive charge, some that have a negative charge, and some that have these crazy rings in here. And all of those things are going to make them behave differently. And again, for our class, remember that you don't have to memorize the chemical structure or even the names of these amino acids. I just want you to know that that R group gives them their unique chemical properties. All right, now we've talked about codons a little bit, and it was just kind of like I hurried past it and we didn't really focus on it. So if we remember, codons are these 3 bases in our RNA sequence that provides the information that tells us which amino acid needs to be incorporated. So it's the sequence of actually the tRNA that's formed off the mRNA that's actually formed off the DNA that tells us which amino acid is going to be coded in. And over years of research, we've actually broken the genetic code, and we can actually know which amino acid, what the codon code, the 3-letter sequence, says to us to go which amino acid to go fetch. And so we'll talk about these in a little bit. But here, you know, here's our codon. UUU is a codon. UAG is a codon, right? So there's 3-letter codes. All right, so there's 64 possible codons in our combinations, but we only have 20 amino acids. Why that ratio? Why those numbers? So let's play this what if game. So if our codons were only 1 letter long, we would only be able to make 4 unique combinations. So we can have the math do this really easy for us. So if we have our 4 bases, A's, C's, G's, and U's, right? And if there's only one possible, if they're just 1 letter long, then we just do 4 to the first power, which is 4 times 1, which is 4. So that's not enough combinations to give us all of our 20 amino acids. So, all right, so that's not going to work. So what if we did 2 letters long? How many possibilities would there be? Well, we can do the same math, use the same formula. So we still have our same 4 bases. So that's going to be our number here. And now we can put our exponent as the number of bases, or how many many bases long our codons are. So if we did 2, then that would be 4 to the second power, which is 4 times 4. That's 16. That's still not enough. So we really have to go and look at it. If it's 3 letters long, then we can say, all right, 4 to the third power is 4 times 4 times 4, which is 64. Boom, there we go. That's more than the number of amino acids that we have. And so that is the minimum. That's the shortest that the codons could possibly be in order to have a unique combination for each amino acid. So that means that we have more unique codon combinations than there are amino acids. So now we have a little bit of wiggle room. We can have some mutations in the genetic code in our DNA or RNA that might not actually change the order of the amino acids, because that a mutation might still code for the same amino acid. So that just means that the genetic code is degenerate, right? So that just means that there's can be mistakes and it's not the end of the world. It's not going to change everything. So if we had a mutation that changed our U to a C, we would still be coding for the same amino acid. Or, you know, here when we're talking about serine, there's all sorts of things. That last position in the codon can have some variability, and it's okay. All right. So let's talk about some special codons. The only codon that you have to remember for me is this AUG, and that is our start codon. That's going to code for the amino acid methionine. And then we also have some of these stop codons, these unique 3 base pair sequence that tells us when we're done. All right, when we hit the stop codon, or sometimes you will hear it referred to as a nonsense codon, it means the same thing. It just means that's the end of making this protein. And then, so then our reading frame is between the start codon and the stop codon. Okay, so let's look at how we can go through. Well, we did transcription now, so now let's talk about translation. And remember, so we had our DNA sequence, and I split this up into its codons just to make it easier for us. We already built our mRNA sequence here. Now we need to make the complement to the mRNA, which is the tRNA. Remember, these are the little pieces of RNA that go into the cytoplasm to fetch the amino acids and bring them back to the ribosome and have those amino acids be incorporated into the new protein chain. Okay, so in order to do that, we just make the complement of our mRNA. So this codon that's CAC is the complement is GUG. Okay, and you can practice doing that. But I've already written them out for you right here. All right. So now what we need to do is figure out how to use this crazy chart down here. This is really confusing at first, but it's really actually okay. So we'll just start here with our first codon. We'll look at GUG. So what we need to do is find on this, on the left side is our first letter. So we need to find, okay, so it's G. So it's not U, not C, not A, it's G. Cool, cool, cool. Now our second letter is, that information is up here on across the top. And so our second letter is U. So we're going to be down here in this little quadrat, this section of this chart. And then our third letter, that information is over here on the right. And so for us, it is GUG. And so now we have the amino acid valine. And that's the first one in this particular little sequence. And then let's just do one more, just because this is kind of intimidating at first. CAC, we're going to go to our first letter, which is C. So it's going to be somewhere along this row. And now we can start to narrow it down a little bit because now we're going to use our second letter, which is A. So we just find where those two things intersect. So it's either going to be histamine or I can't remember what that one is called. But so what we need to do is find our third letter, which is C. So boom, histamine. That's the amino acid that that particular codon codes for. And we can go through and we can figure out what the rest of the sequence is. So the last thing that we need to talk about really is frame shift mutations. So in this example, we have AGC, which codes for a particular amino acid. But here is where we get our mutation. So in this example, this U and A are deleted in this mutant strand over here. And so our first codon is the same Z's as it was in our non-mutated sequence. But now that there's a shift in our codons, and that can really change the protein, as you guys can see. And so instead of valine, now we have alanine. Instead of proline, we have leucine, and etc. So those frame shift mutations are can really be detrimental to how the function of the protein. And then the very last thing that we just need to make sure that everybody's on board with is that the genetic code for protein synthesis is the same across essentially all organisms. And that's why you can see these transgenic mice that glow in the dark. We can take that genetic code that codes for proteins that will glow in the dark, the bioluminescent proteins, and we can we'll take those proteins from, jellyfish or whatever, and we can stick them in mice, and then those mice will have produce proteins that allow them to bioluminesce. And so, of course, with GMO organisms, it does raise some ethical issues that we need to consider. But, this is one of the evidence. This is a big piece of the evidence that says that we had that all life has evolved from a common origin, because we don't have different mechanisms of making proteins, and we just have this one way to do it. So that suggests that we're all related to each other. Okay, so let me know if you have any questions on that, and I will see you guys soon.